Hello, we're glad you've joined us for this live webinar, Validation of Clinical LC-MS/MS Methods, What You Need to Know. I am Judy O'Rourke of Labroots, and I'll be moderating this session. Today's educational web seminar is presented by Labroots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration, and is made possible by an open educational grant from Agilent. Agilent has had no input in the selection of speakers, content, or mode of presentation. Let's get started. You can post questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speaker, Deborah French, Ph.D., the Assistant Director of Chemistry and Director of Mass Spectrometry at the University of California, San Francisco, UCSF Clinical Laboratories. Dr. French received a Ph.D. in biochemistry from the University of Strathclyde in Glasgow, Scotland. She completed a pharmacogenomics postdoctoral fellowship at St. Jude Children's Research Hospital in Memphis, Tennessee, and then completed a clinical chemistry postdoctoral fellowship at UCSF with Alan Wu. In her current role, Dr. French develops and validates mass spectrometry assays for steroid hormones, therapeutic drug monitoring, and toxicology. She teaches clinical laboratory scientists and residents about clinical chemistry and mass spectrometry and oversees the running of the chemistry section with the Director of Chemistry. Dr. French also holds a faculty appointment as an Associate Clinical Professor in the Department of Laboratory Medicine at UCSF. Dr. French will now begin her presentation.
Thank you for that introduction and for the invitation to give this webinar. So I just quickly give you some financial disclosure information. I have received honorarium and expenses from a few places, and I received my salary from UCSF, but none of that's applicable for today's webinar. So after this presentation, you should be able to hopefully write a validation plan and pre-validation recommendations, discuss best practices and solutions for determining common validation parameters, and also write a validation summary and establish some mass spec data review and reporting criteria. So just to give you an overview of what I'm going to talk about today, we're going to cover what method validation is and why we should validate our methods. We'll quickly cover some available resources, and then we'll go into what pre-validation is, and then the majority of the presentation will be on the method validation parameters. We'll talk about post-validation monitoring, and then I'll give you some advice from someone who's probably done it the wrong way more times than she'd care to mention. So what is method validation? It's an objective process that determines that your mass spec method meets the requirements for its intended purpose through laboratory studies. So just imagine that you're performing studies in your lab over a given period of time. You want to make sure that your mass spec method is robust enough so that every time you run the sample, it's going to give you the right result, and that it's reproducible, and that the analyte is stable in the conditions that you're likely to encounter during normal routine analysis. The way the lab does this is by carrying out experiments that address specific analytical variables over a period of time under conditions that mimic everyday use in the laboratory. So you can kind of think about method validation as stress testing your mass spec method. Things to remember are that all methods have error. So it could be random error, systematic error. You can have issues with selectivity. The goal of your method validation is to define the nature and the magnitude of this error. So why should we validate our assays? Well, in some regulatory environments, you are required to validate your method. And these requirements may vary depending upon exactly what function your laboratory has. It's also best practice to validate your method, and the reason for this is that you really want to ensure that your method is accurate, reproducible, and robust, so that it's not affected by small variations in things like new mobile phase or chromatography columns, room temperature fluctuations, or if a different technologist runs the instrument. Think about it, if you're participating in some kind of study over time where a patient is being monitored, for example, you want to know that any change in results is due to some clinical reason and not because of your method. So what are we going to validate? Well, today we're going to discuss the validation of drug X. And you can see a diagram of it here. So one thing to remember is that you are not alone. There are resources out there, and some of them are listed on this slide. So if you're a CAP-inspected lab, the checklist for chemistry and toxicology has some guidance regarding method validation that is general for all methods, but there's also some specific guidance for LC and for mass spec. The Clinical Laboratories Standards Institute, or CLSI, has an improved guideline, C62, that really delves deep into mass spec method validation. There's also other guidance out there, such as the FDA guidance, the EMA guidance, and the European Union directive that all discuss validation of bioanalytical methods. You can also look on PubMed for published papers to see how other people have done validations. But one thing to note is that potentially they're not held to the same high standards that you're held to. So you have to be careful that you definitely do what you need to accomplish. Don't just rely on papers. So if it's your first method validation, you can always ask colleagues that run mass spec methods. We don't bite, I promise. You can also use consultants. I've never done that, but it's definitely something to consider if you don't have a lot of time or experience to dedicate to method development and validation. So one thing that I would definitely recommend is that before you start your validation, that you establish the acceptability criteria for each component. You can find potential acceptability criteria or guidance on acceptability criteria through regulatory requirements, and this could be national or state dependent. You can also use criteria that's noted in the guidance documents that I mentioned earlier from CLSI, the FDA, and the EMEA. But if you're measuring an endogenous analyte, you can also base your criteria on biological variation. And the Westgard website at the bottom of this slide documents biological variation, and it also includes desirable specification for precision and accuracy of the measurements for each analyte. So when you've got to this point, you should have your LC method, your mass spec method, and your sample preparation method sort of ready to go. They should all be sort of set. So pre-validation testing, you can kind of think about it as a mini validation. So it's smaller scale than a full validation, and the point of it really is to help weed out issues before you embark on a full validation. If the data from the pre-validation looks good, then you can go into full validation, which includes determination of a bunch of different parameters that are listed on the slide. And again, as I mentioned, these documents at the bottom of the slide are very good guidance. So what is pre-validation testing? It essentially overlaps with method development. So you develop your method and then you run some samples to test it, and if it doesn't perform the way that you wanted it to perform, you can go back to method development and tweak your method. So that could be the chromatography method or the sample preparation method, depending on what you need. So when you're at this point, you need some kind of blank matrix. So a matrix that does not contain your analyte or analytes of interest. And that's because we're going to do some spiking studies. And also at this point, I usually prepare my own calibrators. Even if I'm going to purchase calibrators later, I would prepare some just for some pre-validation testing. And you need a blank matrix for that. So the acceptability criteria for a blank matrix is that it should have no peak, or if it does have a peak, the peak area should be less than 20% of the analyte lower limit of quantitation and less than 5% of the internal standard peak. And this comes from the CLSI C62 guideline. Now, as you can imagine, this can be challenging if you're analyzing an endogenous compound. You can buy stripped matrix, but it could be very different from your native matrix, for example, serum or plasma. And so it is recommended that you should test as many lots of the stripped matrix as possible because variation is likely. However, that's obviously cost prohibitive. Another option is to use some kind of surrogate matrix, for example, phosphate buffered saline with albumin or water. But one thing to note is that if you are going to make calibrators, for example, in a non-normal matrix or a non-human matrix, that there could be bias introduced because of matrix differences, because as we'll see later, the matrix can run differently in the mass spec. And so a good way to do this is to check the slope of the calibration curve prepared in different materials, or to check that your calibrators have OK concentrations verified by the certified reference material if it's available. You could run human samples or proficiency samples just to check that your calibrators are accurate and that there is no measurement bias. So some of the pre-validation experiments that I would recommend are shown on this slide. So in order to determine if your method is linear, you could run a calibration curve and make sure that you can the method is linear up to the concentration that you would like to achieve. In order to check accuracy, you could run a reference material if it's available. One thing to note is I would run human samples as soon as possible. Your method can look great when you're just spiking your analyte into matrix, or if you're using just standards, but then you run human samples and you see all kinds of different peaks or interferences. So I would run them as soon as possible, because then you can go back to the drawing board and potentially change your chromatography to make your method better. I would test the imprecision of your method. You can use quality control materials to test within run and between run imprecision. It's also a good idea to check your lower limit of quantitation to see if you're getting the sensitivity that you need. And we can do this looking at the precision of the measurements or signal to noise, and we'll get to all of these later. It's also a good idea to test your method for matrix effect and for extraction recovery. And then it's also good to test known interferences. For example, if you want to measure testosterone, DHEA has the same mass, so you want to make sure that you can measure testosterone and DHEA independently. So now we're going to talk about full validation. So for pre-validation, you would probably just do fewer studies. So whatever I'm going to say for validation going forward, for pre-validation, you can do sort of a limited version of full validation. So CLIA has requirements for laboratory developed tests, which most mass spec methods are. And these requirements are that you should determine accuracy, imprecision, analytical sensitivity and specificity, the reportable range, reference intervals. You also should determine your test system calibration and control procedures, and you also have to document all your activities. So the first thing I would recommend is to actually write a validation plan. In this plan, it's good to document all your supplies that you're going to use, the reagents, your quality control material, your calibrators. It's a good idea to have a procedure written at this point. Although, remember your validation, you can also stop your validation if it doesn't look good and go back to the method development part and then start your validation again. So you can tweak these things. It's good to have the samples that you want to run, any proficiency testing samples that you have, your primary standards, because again, we're going to do some spiking studies, so you need your pure standards of your analytes, and then the internal standards that you're going to use. It's a good idea to include the experimental details, so how many samples you're going to run, how many times you're going to run them, over how many days, things like that. And again, you also want to define the acceptance criteria. So on the slide is a suggested order of experiments. Of course, you can do the experiments in whatever order that you want, but it's definitely a good idea to run some of these things early on. So again, I would make sure your linearity is good, your lower limit of quantitation, I would assess accuracy fairly early on and definitely run some human samples, or the samples that you're going to be running once you go live with your method. You should think very carefully about each run that you do to maximize the amount of information that you're getting from these runs. So, for example, we usually use our quality control material for the Imprecision studies, and so you would basically run your quality control material with every run anyway. You would also have your calibrators, so you can look at linearity. And then usually what I would do is add in the lower limit of quantitation sample to every run too, so that you're just optimizing the information. It's also a good idea to think about stability fairly early on. For example, if you do a run and then the samples are already extracted, then you can potentially put them in the refrigerator or the freezer and then run them after a week or a month to see if the samples are still stable. Okay, so now we're going to drill down and look at each parameter individually. So first of all, we're going to talk about accuracy. So accuracy is the evaluation of agreement between LC-MS test results and the true value. So there are guidelines that have acceptability criteria and a guide of how you should perform accuracy experiments. And so the FDA says that you should analyze control material of a known concentration, and the acceptability criteria is imprecision of less than or equal to 15%, and a bias of less than or equal to 15%. The CLSI also has guidelines. So for EP9, they suggest comparison with a higher order reference method using a minimum of 40 samples, which are preferably human, especially if that's what your mass spec method is going to be running normally. However, there's not a reference method available for all analytes, and so that's not always possible. EP15 suggests that you analyze reference materials in 3 to 5 different runs, with each sample measured in duplicate. And again, there's not a certified reference material available for all analytes, and so that's not always possible either. So C62 came up with some different approaches to assessing accuracy. So best practice is really to validate the trueness of your method using a reference of higher order that's listed by the Joint Committee for Traceability in Laboratory Medicine. So best practice is to do a method comparison with your method versus a JCTLM approved reference measurement procedure. Again, however, there's not one of these available for every analyte. They also suggest you use matrix appropriate certified reference materials, but again, that's not always available for every analyte. A third option is to do spike and recovery studies if the reference measurement procedure or the certified reference material is not available. So one thing to note with spike and recovery studies is that it's a good idea to do it blinded. So if you're going to have somebody in your lab make the samples up, so spiking the samples, then I would have someone different in the lab run the actual samples so that you're running them in a blinded fashion so you can compare the spiked value with the measured value. Other approaches are to use accuracy-based proficiency testing samples to check the accuracy of your method. Again, however, they're not always available for every analyte. And finally, an approach is to use a method comparison with another lab using shared samples. And this is pretty much normally what we would do in our laboratory. And so one thing to note is that you should use samples as close to those that will be routinely measured using your mass spec method. So if you're going to run serum, use serum. If you're going to use plasma, urine, urine. And obviously you want to do this for each analyte that you're measuring too. It's recommended that you use a minimum of 40 samples that cover the analytical measurement range of your method, and that these samples should be run on at least 5 different days if you're comparing to other methods. So this is just an example here. We measured our drug eggs with our method, and then in a comparator method with 66 patient samples. And as you can see, the correlation coefficient was 0.9896, and the slope of the line was 1.068. So that indicates that we have a little bit of a positive bias in our method. If we then look on the right-hand side, we can see the percent bias plot, and you can kind of visualize easier here that there is definitely a positive bias. So if we take an average of the bias of all the samples, we come up with a mean percent bias of 6% between us and the comparator method. And so I said earlier you should set your acceptability criteria ahead of time. The problem when you do something like this is that if you're not comparing to a reference measurement procedure or a certified reference material, then you actually don't know who's right. So in this case, I would say a mean bias of 6% between us and the comparator method would be acceptable. So next we're going to talk about imprecision. So imprecision is the evaluation of agreement between repeated analysis of the same sample. In a method validation, we are going to determine our within-run imprecision. In other words, taking the same sample, extracting it multiple times, and running it within the same analytical batch. We're also going to determine between-run imprecision, which is extracting samples over different batches on different days. So the samples you should use for precision studies, again, should be matrix matched to what you're going to normally use when you're running your mass spec method. So, and again, you need to have imprecision studies for each analyte and for each matrix that you're going to be using. So we usually carry out imprecision studies using our quality control sample that we'll be using in our everyday analysis. So in that way, you can use the same between-run precision data to set your acceptable range for your QC sample. It's also possible for you to make your own, so you can make pools of matrix match samples, or you can obviously also buy material in order to do these studies, and the CLSI documents that are helpful are listed at the bottom of this slide. So for acceptability criteria, for within-run imprecision, it's recommended that you run a minimum of 2 different concentrations with 10 replicants in one run or one batch, and the CV should be less than 15%. For between-run imprecision, again, you should run a minimum of 2 different concentrations with a minimum of 20 replicates, preferably over 20 days, and again, the CV should be less than 15%. So for quantitative mass spec methods, we usually always have 3 levels of QC material in our laboratory, and so we do precision studies with the 3 different concentrations. This is just an example of the data that we obtain from our drug X validation. So on the left-hand side, you can see the within-run imprecision. So this is 10 different extractions from the same sample run in one analytical batch. And the mean that we got was 12.46 nanograms per mil with a CV of 2.8%. So that would be acceptable based on our criteria of less than 15%. On the right-hand side, you can see the data from the between-run imprecision. And this was 28 different analytical runs of the same sample extracted each time. And the mean that we got here was 11.86 nanograms per mil with a CV of 5.5%. So again, that would be deemed acceptable based on our criteria. So now we're going to talk about sensitivity. So the sensitivity of your method is depicted by the lower limit of quantitation, or LLOQ. And this is the lowest concentration that can be reproducibly measured. We can determine our sensitivity by imprecision, and with the acceptability criteria of being less than 20% CV. And this is based on using 20 matrix matched replicates over 20 days. You can also determine your sensitivity by signal-to-noise ratio, and we'll get to what that is in a second. But it's recommended that your signal-to-noise ratio should be greater than 20:1 at the lower limit of quantitation, with 10:1 being the minimum signal-to-noise ratio that's acceptable. And you have to also bear in mind that your ion ratios still have to pass, and we're going to come to ion ratios later on in the talk. And some of the guidelines that are helpful can be seen at the bottom of this slide. So often what I'm going to do if I'm trying to determine the lower limit of quantitation of my method using %CV or imprecision is I'll run a low-end concentration curve. So here you can see that we ran a concentration curve from 0 to 10 nanograms per mil for drug X. And you can see down near the bottom that the signal is not really distinguishable from noise of the instrument. You can't really see the signal higher than 0. So we would reckon that the lower limit of quantitation is probably somewhere in this area. What we would do is then make up a sample about that concentration in order to determine the between-run precision of the sample, and then we would run it on a minimum of 20 times on 20 different days. So at this point, if the imprecision is too high, you can increase the concentration of the sample and start the study again. But if the CV is low and you need better sensitivity, then you can always decrease the concentration of the sample and start again. So in this example, we made a sample with a concentration of 0.5 nanograms per mil, and we ran it on 20 different days, and the concentrations that were obtained can be seen in this table. So then what we would do is take the mean of these concentrations, and so it ends up being 0.49 nanograms per mil. The standard deviation is 0.02, and the percent CV is calculated as 4.8%. So in this case, the lower limit of quantitation could be set as 0.4 nanograms per mil because it has a CV of 4.8%, and that's within our acceptability criteria of 20%. So I mentioned earlier that you can use signal to noise for determination of your lower limit of quantitation, and that's shown on this slide. So you can do this in the mass spec software. Essentially what you do is tell the instrument what's the signal, and in this case it's the peak, and what's the noise, which is the baseline here, and it will calculate the signal to noise ratio. So for this sample of drug X at 10 nanograms per mil, the signal to noise ratio was 136 to 1. So as we mentioned earlier, the minimum signal to noise ratio that you're hoping to achieve for your lower limit of quantitation sample would be 20 to 1. So obviously we would know that we could go to a lower concentration for a lower limit of quantitation here. The one thing about signal to noise ratios is that it's very subjective. You tell the instrument where the signal is and where the noise is, and of course you could pick noise that's really flat as opposed to, bumpy, and it's going to give you a higher signal. So I'm more of a fan of determining the imprecision using the running the sample 20 times than using the signal to noise. So in order to determine linearity, we have to carry out this experiment in every matrix that we'll be using, and obviously for every analyte too. So the linearity is the ability of the mass spec method to provide counts per second values that are directly proportional to the concentration of the analyte. And so it's recommended that you don't use serial dilutions to create your material for linearity experiments, and that's because if you make a mistake in one of them, then even a small mistake can create a large bias, and then if you do serial dilutions, you're perpetuating that bias through all of the different samples. In terms of acceptability criteria, one example is shown here, which is using a systematic allowable error of your lower limit of quantitation concentration, or 10%, whichever is larger. You also have to bear in mind that the accuracy or the recovery of your linearity material should be between 80 and 120%. It's best practice to use a minimum of 5 non-zero points and 5 replicates to determine the linearity of your method. And it's common to use 1/x weighting for linearity, and that helps to maintain the accuracy at the low concentration end of your calibration curve. And so in our laboratory, we usually evaluate linearity by running our calibrators. So here you can see an example where you have the assigned concentration of the calibrators and then 3 replicates, the measured concentrations of 3 replicates. And we have to set our acceptability criteria. So here we use 0.49 nanograms per mil, which is our lower limit of quantitation, or 10%. And so if we plot this with the assigned concentrations against the measured concentrations of these 3 replicates of the calibrators, we can see that they look pretty linear on the scatter plot. We can also see that the error was calculated as 0.42 nanograms per mil, or 2.1%, and that's determined to be linear within the acceptability criteria that we set forward in the beginning. So another linearity parameter that we have to look at during our validation is reportable range, and that's the range of concentrations that you're going to report. And so the reportable range includes dilutions of high concentration specimens or specimens with insufficient sample volume, or concentration of low concentration specimens. And so these dilution or concentration procedures must also be validated. And to do this, we usually would spike 5 different aliquots of each blank matrix that you're measuring with specific concentrations of each analyte, and then we verify if the dilution or the concentration procedure works for these aliquots. So in our laboratory right now, we only have dilution procedures. We haven't ever needed to concentrate any analytes. And so in this example, we spiked blank matrix with 5,000 nanograms per mil of drug X, and then we did a 1:4 dilution, which takes it down to 1,250 nanograms per mil, and the imprecision was less than 15%, and so we designated that as acceptable. So in order to dilute the samples, you obviously need a diluent, and so you could use analyte-free matrix if you can find that, or some kind of surrogate matrix, for example, water, and it's water that we use mostly in our lab. And the acceptability criteria are that the accuracy of the dilution or concentration should be within plus or minus 15% of the nominal analyte concentration, and the imprecision should be less than 15%. So that's why we usually do 5 replicants. So the analytical measurement range is essentially the range of concentrations that can be measured directly without doing a dilution or any type of concentration. And for most quantitative mass spec methods, this is going to be from your lower limit of quantitation to the concentration of the highest calibrator. And so in this example, it would be 0.49 to 2,000 nanograms per mil for drug X. So we tend we run our calibrators with every run, and so we tend to have our AMR set as up to the highest, from the limit of quantitation up to the highest calibrator. But if you do want to prove linearity higher than your highest calibrator, then you would need to do calibration verification every 6 months to prove that you're still linear to whatever concentration that you go up to. So one important thing to look at with mass spec methods is specificity or interferences. And these are the ability of the mass spec method to measure solely the analyte or analytes so that it's designed to measure. And this includes the effect of both exogenous and endogenous compounds on the methods. So if you're measuring your analyte in serum, you should also determine the effect of hemolysis, icterus, and lipemia on the method. So for the acceptability criteria, the interference of an exogenous or endogenous compound should have a signal of less than 20% of the lower limit of quantitation of the analyte that you're measuring. And also the interference should cause less than a 15% bias in results. So during method development for mass spec methods, you should look at isobaric compounds and isomeric compounds. And then if you need to, you can alter the chromatography in order to make sure that you're specifically quantifying your analyte or analytes of interest. But I thought it was worth mentioning again here because you should definitely document the compounds that you tested during in your method validation paperwork. These websites at the bottom are useful to search for isobaric compounds, such as endogenous compounds, or commonly used drugs and their metabolites that could potentially cause an interference in your mass spec method. And if you're going to do spiking studies, it's good to spike these compounds into your blank matrix at very high concentrations, higher than you would find physiologically or therapeutically, just so to make sure that they don't affect your assay. So essentially, you would just take your blank matrix, spike the compounds into there, extract the samples as you would normally and run them as unknowns on your mass spec method. And then you can see if there's a peak or any kind of interference at the retention time of your analyte of interest. So this is just an example here that if you have your drug X, which has a molecular weight of 438 grams per mole, and you have an isobar that has the same molecular weight, then you need to chromatographically separate them before they enter the mass spec. And compounds that share common product ions too need to be separated. So as I mentioned earlier, if you're going to measure your analyte in serum or plasma, then you should evaluate hemolysis, icterus, and lipemia and how they affect your mass spec method. It's also really important to test different collection tubes or collection devices on your mass spec method. So if you're only going to accept, for example, a red top tube, then you obviously have to make sure that that doesn't cause any issues in your method. The collection tubes or devices can have an effect on the analyte concentration because it may absorb to the plastic or the gel. Or it's also been shown that some of the components of the plastic or gel may interfere with mass spec methods. And so if you do see an interference with a specific tube type or collection device, then you should probably not use that tube or collection device, and that should definitely be documented in your validation paperwork. So an example of one of the experiments we use for drug X is shown here. So we spiked bilirubin, lipids, and hemoglobin into blank matrix. And you have to remember and take care of the dilution effect. So if you're spiking these things into your blank matrix for the negative sample, you also have to spike in mobile phase or water or something so that the dilution is the same in both situations. And so then after spiking these compounds in, we took the samples, the negative and positive samples, through the extraction like we would normally do, and then the calculated concentration is shown here. So for the bilirubin positive sample, the concentration was 3.8% higher than the negative sample. For the lipids, there was no difference. And for the hemoglobin spiked samples, there was a difference of about 1.4%. So as you can see above, our acceptability criteria are that the interference should cause a less than 15% bias in the results, and all of these situations pass the criteria, so they don't interfere at these concentrations. And this is just an example of how a collection tube can cause an interference. So here, a drug X sample at the top left-hand side was collected in one tube, and as you can see, there's one peak with no interference. However, if you collect the drug X sample in another tube type, you can see all these different peaks, and the drug X peak is kind of swamped by all these different peaks. So as you can see here, it is chromatographically separated, but if your method didn't chromatographically separate these interferences, then you could definitely misquantify the interference as your drug X, which is obviously a problem. So, you know, in this case, since you can chromatographically separate them, you could potentially accept the tube type, but it may make sense just to say that the tube type is not acceptable. Another way of adding a layer of selectivity to your method is to use ion ratio. And this can be seen here on this slide. So if we have our transition for drug X, 438 to 138, this is the peak. And then we have another transition, 438 to 100, and we get this peak. If we take that ratio of the peak area of the first transition divided by the second transition, we get an ion ratio of 1. If we look here on the right-hand side with an unknown sample, you have again the same transition, the 438 to 138 at the top and the 438 to 100 at the bottom. And if you look at the ion ratio for this sample, it's 1.45. And obviously that's very different to the calibrator ion ratio, and you normally would expect that the ion ratios for your samples and for your quality control material are very similar to the calibrators. And here, I don't know if you can see, but there is kind of a bump or a hump on the side of this peak. So that, in this case, this is kind of an obvious interference, and in this situation, I would not report a result for the sample. I would send it to another laboratory to be run on a different method. But it's not always as obvious as that, so ion ratios can really help you detect the unknown unknowns, because no matter how many interferences you test in your method, there's always a possibility that there's something you haven't thought of, and that can really affect the quantitation of your analyte. And back when we were discussing the lower limit of quantitation, I said that the ion ratios have to pass for those samples, and this is what I was talking about. So matrix effects or ion suppression is something that you hear talked about a lot when you're talking about mass spec, and it really is kind of a mass spec specific parameter that you have to validate. And so it's the influence of components in a sample affecting the ionization efficiency and therefore the measurement of concentration of the analyte in the sample. And essentially, it says the thought process is that no two people are the same, right? So no two samples are the same that are going to enter the mass spec. And so if you're using reverse phase chromatography, which a lot of mass spec methods do, you can get ion suppression very early in your chromatographic run caused by polar components like salts, and these elute in the void volume. And another major source of ion suppression in serum or plasma samples is phospholipids. And these are retained on the column and can elute later in the run, but they can also be retained on the column and they can elute any time, like later on in different samples and your calibrators. And so it's very important to document the ion suppression in your method. And so there's a couple different ways of doing this. So first we're going to talk about the way to quantify the matrix effects or ion suppression of your method. And so the best practice is to evaluate 10 different matrix samples. Now 10 does sound a lot, especially if you're talking about endogenous analytes where it's really hard even to find one blank matrix sample. But a regulatory agency has this in their criteria, and so it's probably best that we all do this. And so you basically take your 10 different matrix samples and extract them, and you take mobile phase and extract that and spike the analyte in at the same concentration in each of these 11 samples. And again, if you're using different matrices and different analytes, you have to evaluate matrix effects for each matrix and each analyte in your method. And this paper at the bottom right-hand side here, it gives a very good description of how to do these experiments. And so this is an example of some of the experiments we did for drug X. So we have our mobile phase and then the 10 different matrices, and this is the peak area of drug X in the second column. And the third column is a calculation. So we're basically comparing all our matrices to the mobile phase, because the mobile phase really should have no matrix effect. And so if we have 100% in our percent matrix effects, that means there's no matrix effects. If the number is greater than 100%, it means we have ion enhancement. And if it's less than 100%, it means we have ion suppression in our method. And so usually we kind of take the average of all 10 of these matrices, and here you can see that we have an average of 104.7%, which means that we have about 5% ion enhancement on average in this method, comparing the different matrices. So another way to evaluate matrix effects or ion suppression in your method is to do it qualitatively. And we do this by using a post-column infusion or a T infusion of a standard of your analyte while you inject the extracted samples. And again, you should use 10 different matrix samples. And this basically permits detection of a negative interference. And so I'm no artist, as you can probably tell, but this is basically how you would set up your instrument. So you have your LC pumps. You have your extracted mobile phase and extracted matrix samples in the autosampler. You have your chromatography column. Then you have a T, and this is where you T in your pure analyte standard. So you have a syringe pump containing your pure analyte standard, and it's flowing at a constant rate into the flow of the chromatography column, or the eluent flow of the chromatography column, and this is all going to enter the mass spec. And so this is kind of an example of what you would expect to see. So again, you have constant infusion of your analyte from the syringe pump. And these three examples here, in example A, this is your mobile phase injection. And so as you can imagine, if you have constant infusion of your analyte from the syringe pump and there's no matrix effects, then you would have basically sort of a flat signal of your analyte. If you inject serum that has been extracted using a liquid-liquid extraction, you can see here that there's ion suppression just before 1 minute of the chromatography run, and then it kind of goes flat, and then there's a little dip here just before 5 minutes, and then it's kind of flat again. So there's definitely some matrix effects or ion suppression in this method right around just before 1 minute. So obviously you wouldn't want your analyte to elute from the column at that time because the signal is going to be very suppressed, especially if you're looking to get high sensitivity from your method. And the third example is if you use a protein precipitation method to extract your serum. And here you can see that there's a few dips in the signal indicating areas of ion suppression. So again, you wouldn't want your analyte to elute from the column in any of these areas. And another thing that this shows is that liquid extraction cleans up your samples much better than protein precipitation, which is something we probably all knew. Anyway, so this is a way to qualitatively see where areas of ion suppression are in your method. Another thing you could do is do the same experiment with your internal standard, and if the internal standard signal dips where the analyte dips, then you can, it kind of shows that your internal standard is kind of accounting for the ion suppression that hits both your internal standard and your analyte. You can also do the same experiment for the quantitative experiment with your internal standard as well. And so shown on this slide are just some references as far as matrix effects are concerned. So another thing that we should look at during our method validation is stability, and that's somewhat difficult to accomplish during validation. You should really test your analyte in the matrix at every condition that it might encounter. You can also do a literature search in PubMed to see if anyone else has already done it, but they may not have used the same matrix or the same temperature that you're looking to store your analyte at, so it's not always successful. You know, if you think about it, if you make your own calibrators, then how do you know how stable they are until you test them for a long time? But that's not really something you're going to be able to accomplish during validation. So the acceptability criteria for stability studies are that there should be a less than 15% change from freshly obtained or freshly extracted sample, except at the lower limit of quantitation, where a less than 20% change is acceptable. So some of the conditions that we test for in our laboratory are how long will the samples be stored before analysis and at what temperature. If you have already extracted the samples but your mass spec goes down, how long can these samples be stored and at what temperature? What if your sample needs to be run at a later date? Again, how long can it be stored and at what temperature? And another thing to consider is freeze-thaw stability. So if you have calibrators and you have, you know, a milliliter, but you only need to use 50 microliters for each run, can you freeze-thaw the calibrators, or do you have to thaw them one time and just use them and keep them in the refrigerator? And you can also think about this for your quality control samples and your human sample. This is an example of one of our stability experiments. So what we did here was take 10 patient samples and we extracted them fresh and ran them on the instrument on 10/30/2017, and these are the concentrations that we got. We then took these extracted samples and stored them at 4 degrees C for 1 week, and then we re-injected these samples a week later, and these are the concentrations that we got here in column 3. And then in column 4, we calculated the percent recovery by taking the result from 1106, dividing it by the result from 1030, and multiplying by 100. And as you can see, the lowest recovery that we have here is 88%, and so that's within the stability, the acceptability criteria of less than 15%. And so it was determined that the samples for drug X are stable for 1 week at 4 degrees C once they've been extracted. So your LC-MS system is a continuous flow system, and so there is the potential for high concentration samples to carry over into the next sample. The acceptability criteria for carryover studies are that it should not significantly impact the bias or the precision of the method. So in our laboratory, we use 1% as criteria for acceptability for carryover. So an example experiment that you could use here is to extract 3 low concentration samples, then extract a very high concentration sample, then extract 3 more low concentration samples and run them in that order. And so what you would do is compare the low concentration samples that were injected before versus injected after the high concentration sample. And this is an example of the experiment for drug X. So we have our 3 low concentration samples, and when we injected them before the high concentration sample, we got concentrations of 12.2, 11.7, and 11.5, so the average was 11.8. We then injected the high concentration sample, which was 10,000 nanograms per mil, and then we ran 3 low concentration samples after the high concentration sample. And so these concentrations of the low samples came out as 12.8, 11.8, and 11.7 for an average of 12.1 nanograms per mil. So then we can calculate the percent carryover by taking the average concentration after minus the average concentration of the low samples before, dividing it by the concentration of the high sample and multiplying by 100. And in this case, we get carryover of about 0.03%, which is within our criteria of 1%. The reference intervals are something that we have to think about, especially for endogenous analytes. And again, these are somewhat difficult to accomplish depending on the analyte. If you want to establish reference interval of your own, then it requires a minimum of 120 normal samples. And another way to do it is to verify and establish reference interval for your population. And in that case, it would require a minimum of 20 normal samples. And the acceptability criteria to verify and establish reference interval is that a maximum of 2 samples could be out with that reference interval. And one thing to remember is that if you have gender-specific or age-specific reference intervals, then you have to verify each one. So you would need 20 samples for each population. If you're looking at exogenous non-therapeutic compounds, then the reference interval would be negative or none present. And then for exogenous therapeutic compounds, if therapeutic ranges are already established, then they can be found in the literature or potentially in the package insert of the drug. Another option is also, if you can, to communicate with pharmacists in your institution, and they can probably help guide you in the right direction. So some other validation parameters that we have to think about are the maximum batch size that you're going to run. So if you're running in vials, how many samples can you have in one run? Or if you're running in plates. You can also document the extraction recovery. So you can spike your sample and take it all the way through extraction, and then spike a sample after it's been extracted to see and compare the difference between before and after to see how much of your analyte you recover, or basically how much of your analyte you lose during the extraction process. You can look at retention time stability, and this is helpful too if you change columns to make sure that the retention time is stable. You can look at the slope of your calibration curve. If proficiency testing samples are available, then you can run them and see how you can compare. If you're going to validate your method on 2 different instruments, then you have to do a between-instrument correlation as well as a full validation on both instruments. You can also document lot-to-lot variability in mobile phase, so you can make up different batches of mobile phase and have different people make them up and then test samples that have already been run. And then this way you can come up with criteria for acceptability once you actually go live with your method. There's probably other parameters that I'm forgetting, but this is a good list. Another thing that you want to do is set up your acceptability criteria for your run. So once you're actually live with your method, what has to happen or what has to be true before you will actually report results. And so you can use retention time of your analyte and internal standard. You can make sure they match. We're using normally isotopically labeled internal standards, so the retention time of the analyte and the internal standard should be the same. You can make sure there's no interfering peaks in the method. You can look at the calibrator recovery or accuracy, and you can set criteria as 80 to 120% recovery. You can set the R² of your calibration curve to be greater than 0.99. Your quality control material has to be in range, so you have to have determined what the range is, and then it has to be in range before you can report results. The ion ratios of your unknowns and of your quality control material should be within plus or minus 20% of the calibrators in the same run or the same batch. And also, you should monitor internal standard peak areas, and these should be consistent between all of the samples too. So an example of criteria that you could use is that the internal standard peak areas in your human samples should be within plus or minus 2 SD of that found in your calibrators. And these are all things that you can use your validation data to determine, right? So you see how your assay performs during validation, and you can use that to set criteria for when you go live, because you want to be able to catch issues, but you also don't want to fail every single run that you have. So it's really important to do a really good validation, very in-depth and over a significant period of time, so that you know how your assay performs on its best day and on its worst day. So once you've actually gone live with your method, you've put all this validation to work, you've gone live with your method, there's things that you should do in terms of post-validation monitoring to ensure that your instrument is performing, that your assay is performing now that you're running it live. And so things that you can use to monitor instrument performance are your system suitability sample, and we'll get to that in a minute. In order to monitor sample analysis performance, you're using your quality control materials with every run. You can use your internal standard peak areas across your run as well. In terms of looking at your assay performance over time, you're going to do proficiency testing. You could do accuracy assessments using a certified reference material if that's available for your analyte. You should do lot-to-lot validation of your mobile phases, your calibrators, your quality controls, your column. And also, if you do run your method on different instruments, then you should do instrument comparisons every 6 months to make sure that they're still running the same way. You should also obviously do instrument maintenance, and this really depends on your manufacturer recommendations, your personal experience, also how much you actually run your instrument. So right now, for example, we clean the front end of our instrument once a month, but if we were to increase the volume of samples that we run, or if we were to run samples that weren't extracted so well, so perhaps samples that we did just a protein precipitation for, then we may have to consider cleaning the front end of the instrument more often. We do have the manufacturer come in and do periodic maintenance every 6 months as well. So what's your system suitability sample? So it's essentially a non-extracted sample. It does not go through sample preparation, and so this is really just a test of your instrument. It's not a test of your sample prep process. It's a test of your instrument on the day that you're running. Some different concentration suggestions are that you should have it near the lower limit of quantitation to evaluate your instrument sensitivity. You could have it in the middle of your analytical measurement range, or near a medical decision point. So basically what you do is prepare a large batch of this. So it's just your analyte spiked into, for example, methanol and water or something like that, and you have to store it according to stability standards. So if you've validated stability, then you know how to store it. You essentially want to run this system suitability sample daily before you do any analysis to make sure that your instrument is performing the way it should do. After any instrument maintenance or a power outage or any kind of break in vacuum of the instrument, and also after you've had periodic maintenance, like the tuning and calibration has been carried out, because you basically want to make sure that the instrument is performing the way that you know it should. And so parameters that you can monitor for the system suitability sample are a shift in retention time. This could be gradual or sudden. And there's a number of different reasons that the retention time could shift, and they're seen here. It could be the column degrading. It could be that your LC buffers are evaporating, or there's been some kind of pH change. It could be that your LC pumps are not mixing properly, or there's a leak somewhere. You can also look at the peak shape of your system suitability sample. So is there any tailing or fronting, or is the peak asymmetric? And again, this could be indicative that there's something wrong with your column or your buffers or your mass spec itself. And again, if there's any additional peaks observed that are not usually observed, there could be a problem. Also, if there's a drop in intensity of the peak for your system suitability sample, it could be due to your mass spec needing cleaned, again, changes in your buffers, or potentially something wrong with the injector on your system. It could be clogged, or the loop could be clogged, and it also could indicate column degradation. So it's really important to monitor system suitability sample every time you run it and to document what's found. And so then you could potentially go back to and see if over time there has been a gradual decrease in the peak intensity. So is that your instrument getting dirty, or is the actual system suitability sample not stable? So now that you've done all this hard work it's time to write your validation summary report. So usually the lab director signs off on kind of a one-page summary, but that's not really very informative. So it's helpful to put together like a validation summary that summarizes the results for each experiment that we've just discussed. And this is basically the meat in the sandwich. And you can also, at the bottom of the sheet of paper, put the electronic file location. And that's very useful because then you can go back and find it much more quickly. If there was any failures during the method validation process, but that you fixed them, then put that in there. Don't omit data that revealed some kind of true problem that you fixed. And also include corrective action. So if you found an error that could have clinical significance, for example, some kind of interference at a very high concentration of a drug causes the ion ratio to fail, write that in there, and then you would include a comment about the interference in the validation report. So now that we've validated our assay, this could be like our one-page summary. So for drug X, our accuracy was acceptable. We had a mean bias of 6% compared to another lab. Our lower limit of quantitation is 0.4 nanograms per mil. Our analytical measurement range is 0.49 to 2,000 nanograms per mil. We had acceptable imprecision both within day and between day, and we had about 5% ion enhancement as far as matrix effects are concerned. So lastly, just some advice. I have done this for a few assays now, and every time I do something silly. And one thing that I did that was really silly was do most of the validation myself for one assay, and that came back to bite me as soon as we went live, and we had multiple technologists. So during the validation, you want to throw in every single type of change in condition that you can think of. So you want to have multiple technologists. You want to have multiple mobile phase loads made by multiple different people. If you can, it's good to try different column lots. Obviously, columns are fairly expensive, so that may or may not be possible. You definitely want to carry out normal instrument maintenance. We were in the middle of validating an assay, and we had our preventative maintenance done. And after the engineer left, our assay did not work at all. So it's good to know these things ahead of time. You want to test on sunny days and rainy days. And of course, that's kind of a joke, but it's true. Anything that could possibly affect your assay, you want to throw at it during method validation so that when you're actually live with it, there's less chance of these things happening that could affect the performance of your assay. And lastly, document, document, document. In our lab, if it's not written down, it never happens. So document every little thing. And this, again, could be during validation. This could be afterwards. You know, any troubleshooting that you do, write it down. Write down what the problem was. Write down what you did to fix it, so that people can go back and refer to it in case the same problem happens again. And so in conclusion, per Dr. Russell Grant, validation is a risk-based exercise. The more work you put into a validation, the less risk you have of your method failing afterward. I would just like to quickly acknowledge some people who helped put this presentation together: Dr. Judy Stone, Dr. Josh Hayden, Dr. Julie Botello, and then everybody at UCSF Clinical Laboratories who I work with. And with that, I would like to thank you for your attention, and I would be happy to take any questions.
Thank you, Dr. French, for your presentation. Quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question, and click on the Send button. Dr. French will answer as many questions as time permits. Okay, the first question is, for pre-validation, do you need to use your finalized calibrators and quality control?
So not necessarily. I think I mentioned earlier that I often will make my own calibrators during pre-validation just to test the method, just to make sure that they work. So, you know, if you have already purchased calibrators and then you find out that your assay isn't linear to the concentration, the highest concentration, then, you know, that would be terrible. So I would say it's probably better to test your system first to see what linearity you can achieve, to make sure that your calibrators cover the linearity that you want, and then decide where your quality control samples should be, and then potentially order them or make them after you've sort of done your pre-validation. So once you're more happy with the exact measurement range that you're going to use.
Thank you. Your next one is, can you do between-run precision experiments over 5 days, 4 replicates per day?
Yeah, so this is a common question. You can. I think the requirement is 20 points. I just wouldn't recommend it, and the reason for that is, like I kind of was drilling home at the end, you really want to throw as many different things at your method as you possibly can. And if you only validate Imprecision over 5 days and you do 4 replicates, you're not really getting the variation of different days, different weeks, different people, right? I mean, I guess you could get different people over 5 days. Again, it's a risk-based exercise. The more you throw at it, the more chance you have of seeing it fail at this point as opposed to later on. And so I just think you get a better idea of how your method is going to perform when you run it over more days. And the other thing to remember is you have a lot of experiments to do during your validation. You have patient samples to run, you have your precision samples, you have carryover, you have reportable range. You have a lot of studies, so really you are going to do a lot of runs. And so it's not really an added hardship to do precision experiments over more days than just 5 days at 4 replicates per day.
Thank you. What material do you use for the hemolysis, icterus, and lipemia experiments?
So that's a good question. It's a little controversial for lipemia because we usually do use intralipid. So you can buy intralipid. It's what people use for TPN and hospitals, so patients that can't eat, they get intralipid intravenously. It's not really a good capitulation of what you will find in a lipemic sample, but it's kind of a surrogate. So we use intralipid for lipemia experiments, so we just measure the lipids in that using our chemistry analyzer and then get the concentration, and then we can figure out what we need to add to our samples, our spiking samples. For icterus, we actually have been using calibration verification material just to spike, that contains bilirubin to spike our samples with. And for hemolysis, what we actually do is lyse red blood cells and then measure the hemoglobin, and then again spike into our samples using the lysed red blood cell.
Thank you. We have time for one more question. How do you find blank matrix for ion suppression experiments of endogenous analytes?
Yeah, so that's a really good question. You can use stripped matrix, so you can buy double charcoal stripped matrix to do these experiments. However, with the regulatory requirement of 10 different matrices, that makes it kind of difficult. So what we tend to try and do is look for patient samples that either have a really low concentration or like undetectable concentration, which can be very difficult, but we do get them. And then we can use them for spiking studies, for the ion suppression studies, so that to spike your analyte at a certain concentration. The other thing you can do is use patient samples and run them straight, first of all, to get the actual concentration of your analyte. And then when you add your analyte on top, at a certain concentration, then you can just sort of subtract the concentration that you found when you ran it straight before you were doing the ion suppression experiment. So we just did this for estradiol, and so we thought about picking samples that were kind of potentially low estradiol concentrations. So in that case, we looked for adult male samples and then for some pediatric samples, and we ran them straight first so that we got like a baseline concentration, and then we spiked on top a known concentration, and then we just subtracted the peak area out of there. And so you don't necessarily have to use blank matrix, and I feel more comfortable using real patient matrix rather than the double charcoal strip matrix, because it definitely is a better indicator of what's going to happen with your real patient sample.
Good. I would like to once again thank Dr. French for her presentation. Do you have any final comments?
No, just thank you. Thanks again for the invitation. And I don't know if the slide is still up, but it has my email address on it. If anyone has any further questions, then they can feel free to email me.
Okay, thank you. I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our educational sponsor, Agilent, for today's educational webcast. This webcast can be viewed on demand through March 2019. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.